We will be discussing the importance and process of gene regulation in prokaryotic cells. This is a big topic, and there is a lot where our talk could go into, even when just considering the various aspects of practical implications. But today we will be focusing on the main concepts. I want to start by taking a step back and quickly reviewing the basic concepts and terminology of genetics, and what makes prokaryotes different from eukaryotes. I also want us to put some numbers to this process to illustrate the importance of gene regulation. Next, I have decided that we will tackle the topic through looking at few real-world examples by gene regulation in a well-studied prokaryotic model organism. Finally, rather than just giving you a single answer for why the gene regulation is important in prokaryotes, I hope that the answer will become clearer throughout the talk as we discuss the process. Let's start by reminding ourselves that most activities of an organism are controlled by genes, and nearly all cells of an organism contain all the genes that individual has. This is what we call organism's genome. I like to compare genome to a cooking book. We have genes, which are like recipes, and nearly every cell contains all of these recipes. However, we do not want all of them actively working at one time, just as we do not want to be cooking every single recipe of a cookbook at one time. Because if all the genes would be functioning at all the time, at all the cells, we would end up with a state of a complete chaos. Therefore, a cell will typically only activate a gene when the product encoded by the gene is needed. This product is often a protein, which, by carrying on with our analog of cooking, would be the final dish. So we do not want to be making all the possible dishes that we know all at the same time. That would be just silly. So most of the time, genes are shut off. This turning genes on and off is called gene regulation. During this talk, we will be referring a number of times to a well-studied prokaryotic model organism, E. coli. Taking that organism as an example, just to give us some perspective, at any given time, only some of its about 3,000 genes are being expressed. In a typical human cell, only about 20% of the genes are expressed at any given time. Let's remind ourselves here quickly about the key characteristics of prokaryotic cells. These are single-celled organelles that lack nucleus, and other membrane-enclosed organelles. Therefore, DNA floats around free in the cytoplasm of a prokaryotic cell. By this stage, I believe that you should have covered the process of transcription and translation as a mechanism of producing a functional protein. Because prokaryotes' cell structure, unlike in eukaryotes where we have distinct compartments, the process of transcription and translation happen almost simultaneously. Therefore, in prokaryotes, we mostly use the regulation of transcription to control what protein and how much of it to express. So I guess that this begins to answer our question of why gene regulation is important. We will keep adding more to this answer as we move ahead by looking at how this process actually happens in E. coli. So we remember that prokaryotes only transcribe genes when the end product, protein, is needed at that very time. They do this to save energy and increase their efficiency. We remember that prokaryotes are fairly simple organisms, so they, or their cells, do not have a brain to think about, what genes should I be now expressing? Instead, prokaryotes respond to the changes in their immediate environment by regulating their gene expression. A good example of this would be whether nutrients are present or absent in the medium where a prokaryotic cell happens to be at. So we talk about extracellular signals that affect prokaryotes' gene regulation. So these extracellular signals that I mentioned are carried to the genes by regulatory proteins. Before we look at regulatory proteins, I want to introduce what I'm showing on this slide. This is just a short segment of prokaryotes or E. coli's DNA. Often in a bacteria, we have structural genes that are functionally linked. In this example, it would be structural genes Z, Y, and A that produce enzymes that function all together in same part of a metabolic pathway. Since these genes work all together, they are also found in the DNA as a cluster next to each other. What is even more impressive about this clever design is that because they all are needed together, their transcription is controlled by same regulatory genes, I, P, and O. We call this bundle as an operon. We will be looking at a regulatory protein. Remember, they carried extracellular signals to the genes. Using one well-studied example of E. coli's operon, the Lac Operon. In this case, Lac stands for lactose. So let's take one step backward and think, why lactose? Well, you might not consider lactose as a great meal, especially as many of us are intolerant to this sugar found in milk. But E. coli certainly thinks that it makes a great meal. Ideally, E. coli would prefer to consume sugars that are most easily digestible. This can be easily split into small parts, such as glucose, which is a monosaccharide. But if simpler sugars are not around, E. coli uses the next most simple form of energy, such as lactose, which is a disaccharide that takes a bit more effort to split into smaller parts. Lac Operon works efficiently only when glucose is not available and lactose is available in E. coli's environment. We check for the presence of these two by using specific mechanisms. We will start by discussing about lac repressor, which functions as our lactose sensor. Under normal conditions where lactose is not around, regulator I gene produces a protein called repressor. Repressor basically inhibits the expression of structural genes. In a normal situation, this repressor is active and binds to the operator O gene. For transcription to happen, RNA polymerase needs to bind to promoter P gene region, but this is now prevented because there is overlap between the binding sites of promoter and operator. This is a perfect example of how repressor protein turns genes off in what is known as negative control. However, if we do have lactose present in the immediate environment of E. coli, a molecule called inducer, this is activator, is induced from the outside of the cell. In this case, it would be allolactose, which is another form, this is isomer, that lactose takes when entering into E. coli. Allolactose acts with the repressor, changing its shape, which makes repressor inactive and unable to bind to the operator O gene. Now promoter P gene is free, allowing the RNA polymerase to bind and the transcription to proceed. Proteins encoded by the structural genes Z, Y, and A are produced, allowing lactose to be metabolized and processed, broken into small parts, in the prokaryotic cell. Therefore, this process is an example of how the presence of a specific substrate, in this case lactose, causes increased expression of the target gene. This can be also known as induction. To illustrate the effectiveness of this process in the absence of lactose, E. coli has on average only 3 beta-galactidase enzymes produced by LacZ, whereas following induction of lactose the number increases to about 3,000 within just 2 to 3 minutes. As I mentioned at the very beginning, that for Lac Operon to work optimally, both of the two conditions must be fulfilled. Lactose must be available, we just confirmed this, and glucose must not be available. This control mechanism allows us to maximize the transcription of Lac Operon when glucose is not available. We use catabolite activator protein CAP, which functions as our glucose sensor. If glucose is not available, cyclic AMP, cAMP is produced. It joins with CAP, forming cAMP CAP complex. This complex binds to regulatory CAP site on DNA, further activating and promoting RNA polymerase to transcribe the structural genes even more. If glucose is available and being used, a breakdown product of the glucose, called glucose catabolite, is produced. This inhibits the production of cyclic AMP, resulting in lower levels of cAMP. Although some amount of transcription happens, remember earlier Lac Operon's function, CAP does not get activated and bind to the CAP site. Consequently, the transcription of Lac Operon remains low. In this case, cAMP-CRP complex is added to the story by enabling high or low levels of transcription of Lac Operon. Having both control mechanisms to activate Lac Operon, a negative control by inactivating the repressor, and positive control, which we just introduced as cAMP-CRP complex, allows E. coli to adapt more efficiently to the changes in the environment of its natural habitat, which is often the human intestine. Finally, if we have time, I would like to introduce another example as a revision. Also, Trp Operon can be found in E. coli. Trp stands for tryptophan, and it is an amino acid that is synthesizing proteins. The big difference in comparison to Lac Operon, which we saw earlier on, is that under normal conditions, no or low tryptophan, the repression protein is inactive and tryptophan generating genes are expressed, turned on. But when the end product tryptophan is produced, it acts as a co-repressor by binding to the repressor, and now the repressor is activated. Consequently, the activated repressor binds to the Operon O gene and blocks the transcription. Gene is not expressed. To put it simply, if we are in an environment where we have lots of tryptophan, there is no point of encoding for making more of it. But when there is no tryptophan, it makes sense to make some of it. This example explains how bacteria are able to synthesize enzymes and stop their synthesis as soon as they have been produced. We have now came to the end of our journey in the prokaryotic cell E. coli. I think that we should now have a good idea of the importance and mechanisms of gene regulation in prokaryotic cell. Looking back at what we set out to do, I believe that we have covered the basic terminology in genetics, discussed the key processes using real-world examples, and hopefully walk away with an appreciation of the importance of this process.